we are shifting to the next part. So now we have the basic background about a cell. So what we're going to do, we are going to go further details about a cell. So there's an in this tube I just put, there's a small experiment to do. So you are going to observe animal cells. So let's see how we observe animal cells in this video clip.
Neat place, Jasmine.
The journey into a drop of water begins with a visit to your local pond. These students are collecting pond water to study later at school. They start by collecting some pond scum. This is usually filled with microorganisms. Then they add water. Finally, they cap their jar. There's enough air in the water to keep the organisms alive for a day or two. They'll also collect water from several spots. Back in the classroom, an eyedropper is used to deposit a drop of water onto a glass slide. The slide is then covered with a thin piece of glass called a cover slip. The slide is then placed on a microscope stage beneath two clips. The organisms you see in a drop of pond water may look strange, but they share much in common with you and me. Here's how you can prove it. Scrape the inside of your cheek with a toothpick. Dab this on a glass slide and cover it with a cover slip. Then place this under your microscope. As you can see, the inside of your mouth is made up of tiny uniform structures called cells. A cell is the basic unit of which all living things are composed. A person's entire body is made up of cells. Billions of cells that are not that much different from the ones that make up the organisms you find in a drop of water.
Hope you got the massive idea how to produce a special specimen as well as observing it in the low power and all these things. So in here, in this space, you can do it, you can practice this, write the steps, main steps, nearly 5 steps are there. First step I will recall again. First thing we are going to say, take a small sample of living cells. For example, we took animal cells from our cheek cells, because assume that we are also animals actually. So we take some cells from your cheek cells, cheek epithelial of the surface by using a toothpick. Install that or put that sample onto a clean glass slide, add one drop of water and cover it with a cover slip. Then what we do, we observe the specimen. Now we call it a specimen. We observe the specimen under low power. Remember that thoroughly, under low power of the compound light microscope. Then if we want to focus it and see further details, we go to mid power and finally to high power. Then that's the procedure for observing animal cells. Then in here, what you observe, you have to draw. That's another skill you have to practice, drawing the observation. You are doing that thing in this method because that method you have to remember for the rest of your life, because if you do science for advanced level also, you need to draw figures, what you see from the microscope, through the microscope. That means it's a practical idea. So when you draw this thing, you have to take a pencil, and that pencil should draw a pencil line diagram, not a drawing actually. We don't draw nice arts here. We are drawing a diagram here. In that diagram, it should be line diagram. The boundaries of the cells are marked, and the few structures that you see inside the cell should be marked as you can see that from the microscope. And from that thing you can take a conclusion, what can we observe from this thing. You can observe there are cells in an irregular shape, and these cells have a central structure. Still we don't know what is that structure. You can see here that the cells in the living animal are not having a proper shape. The shape can be varied, right? That's the one thing you can observe. And there's a small central structure you can see as a dot. We will know that what is that thing in the future in this session. Still remember the name, just remember the name. So that is the nucleus of the cell. So those are the things we can see. Now same type of experiment what we do here. We are going to learn about the plant cell. This experiment is also the same steps you have, but the specimen we take is the rohio plant. Most of the time we are taking that plant, because that plant, what we do here is, I have to say this thing, normally these cells, most of the cells are transparent. That means they are colorless, you can't contrastly see them. So what we're going to do is we are putting some ink, we call them stains. A stain is put inside the specimen to see the cells or to visualize the cells in a contrasting manner, because otherwise it is transparent. So what we do here, we are taking a stain cell. That means these cells in the rohio plant is a purple color leaf containing bushy plant. In that plant you can see already the cells are colored with purple color. If you can see now how we do the experiment.
Right, my dear students, so today we are observing the plant cells. I told you about a plant, that is the rohio plant. So we can find rohio plant like this because you have to know what is the plant rohio. So it's a plant, you can see here. In this plant, it's a bushy plant. So what you're going to do, we take a sample from this plant. So we take a matured leaf, we select a matured leaf like this. So you can see the lower epidermis, I mean behind the lower epidermis you can see the purple color stain. So what we're going to do here is we are going to make a sample from this specimen to see the plant cells. Let's go. For the preparation of the raw specimen, so what we're going to do here is we are going to need some stuff. So I'll collect them and come right back. For the preparation for that, we need the structure. You know this what it is. This is the light microscope. So it's a compound light microscope. The reason is, you can see in here there's the eyepiece lens and this is the objective lenses. There are 3 objective lenses and the nose piece you can rotate that. So you can understand why we call it a compound light microscope. The reason is you have 2 lenses, 2 or more lenses that will give a huge magnification. And this is the mirror. So from this mirror what we can do, we can reflect the light. That's why we call this the light microscope. So let's see how we prepare this. We need some stuff. This is water bottle. We need a petri dish containing the sample, and we need cover slips. I'll take one slip and see. You can see that. This is we call the cover slip. So this cover slip is used to cover the specimen without contacting directly with the lens, because otherwise when the water is present, so we are adding water to this slide. This is called the slide. So we are adding water. So if the water touches this lens, the lens start to get damaged, because fungal growth will be happen due to the moisture, right? Okay, let's see what we're going to do. And we need a sharp blade. Here you can see the sharp blade, a surgical blade I use here. And we need a slide, a clean glass slide. You can see that, right? We need a petri dish and we need water in that, because these plant cells, if you keep those plant specimen, the tissue what we taken from this in a dry area, what will happen, the cells start to shrink and the cells start to die. So we need to see healthy cells. So ultimately after you take the specimen, what you're going to do, we are going to put those samples onto the water. Right. Now we are going to take a thin specimen in this leaf. This is called the rohio plant. The reason why we use this plant, I will recall that, because normally any animal cell or plant cell, sometimes they have no color. They are colorless, that means transparent. So you can't see those tissues or the cells very clearly using the microscope. So what we're going to do, we are going to make these cells colored, but that process is not considered up to the ordinary level. So what we do, we use cells which are colored in this. So that's why the purple colored cells we are taking from the rohio plant. So let's see how we take the cut. You can turn this leaf a little to make a curvature like this. Then you can take a thin specimen. So specimen should be very thin, otherwise so many layers of cells will cause transfer of light, so you can't see the cells clearly. So what you're going to do here, you can see. And the other thing, you are not cutting the specimen towards outwards, you're cutting the specimen towards yourself, then you can control your hand, because the hand is moving up to a certain limit. You can take multiple specimens. Sometimes you have to take a sharp blade here. You can take multiple specimens like this and put immediately into water. So you can take some more specimens to get the thinnest one, right? So we prepared the specimen, right? So what you're going to do here is we are going to take that specimen onto the glass slide. So to take the specimen, normally we use a paintbrush. So I don't have a paintbrush in this situation, so I'm using a pencil. So you can see here, what you're going to do here is we are taking the specimen onto the glass slide. Specimen will stick because of the surface tension of water, so you take the specimen and you are transferring onto the glass slide like this. So you can see the specimen now like this. It's a large specimen, you can see we have taken here. Now what we're going to do here is we're adding a water droplet here. Right, now what we're going to do here is, after adding a drop of water using the cover slip, you're going to cover the specimen. After covering the specimen, you will see, and if there are excess water in this area, so you can blot them out using a tissue paper. So what we're going to do here is we are going to mount this into the light microscope and see under low power. So let's see what is low power here. In this microscope you can see there are multiple objective lenses, and this one is called the eyepiece lens, and these are called the objective lenses. So these lenses have different magnification powers. So start from the low power. Low power is in here. Low power is the red color. And if you want to magnify and see further details of the specimen, you go to mid power. If you want to see further magnified, you go to high power. So the color is not standard here. So in this category of microscopes, you have these color ranges to say that low power, mid power, and high power. Now we are placing the specimen. The object what we are observing should be in this cavity. This is called the aperture. It should be at center, and stage clips will clip that out like this. Now we're going to observe this thing. So remember again what power we use low power magnification for the first time. And here there are two adjustments. I will recall what you have learned in grade 9. This is called the coarse adjustment, and this is called the fine adjustment. So from the coarse adjustments, first we are going to focus the image, then we are finding the image quality using the fine adjustment. Don't use this directly at the first time. So let's see what we're going to do. See, without light it will be not fine and focused. So when we move the mirror, you can see it will get the light like this, and still it is not focused yet. So what we're going to do, we need to focus it. You can see now when you focus this thing, it becomes clear, and you can see polygonal shaped cells, something that similar to Robert Koch's finding. So he found Koch cells in plant epidermis of the bark of a plant. So like that you can see small square or polygonal shaped structure. So one of that is called a cell. If you want to magnify further and see in more details, you can go to mid power. Now you can go to mid power. I'm shifting to mid power. I'm going to fine focus this thing for a small amount. When you focus it, you can see the cells nicely. So one of these polygon is a one single cell. In this, some cells you can see in dark color and they have a central area. So those are called stomata. Those are the structures where these plant leaves breathe, taking carbon dioxide and releasing oxygen gas, right? So can you see any nuclei present? No, right? The reason is they are not that much of visible up to this level of magnification, but you can see clearly the cell wall, the boundary, you can see very clearly. So we are going to draw such a picture in that space given in the tutorial. So that is the way you are observing. You can observe this thing and draw a picture about this. I mean, not actually at art we are drawing a pencil diagram. Let's see what we can see from the high power. When I shift to high power, you can see blurred image. So we are going to focus this thing, fine focus. Now you can see separate cells. See that? So even here you can see the cell wall clearly, and the cell nuclei is not visible up to this level, but some structures you can see, we can identify them clearly in this. This central structure is called the stomata, containing two guard cells. These guard cells, you can see green color circles, spheres, those are called chlorophyll inside. So I will show you some other chlorophyll structure containing cells in here. I saw some in this cell. I will focus it. So you can see now spherical structures in the cell. So these are chloroplast, the organelle that is doing photosynthesis. So that's why the leaf is green in color. You can see them very nicely in this. Hope you got the idea of how to use the microscope. So, but remember, it's a very expensive gadget, so you are going to take it in care.
Now you can see what you observed. You're going to draw that. Don't color it using purple color. Color you can remember, but you are drawing a pencil diagram. What you can observe in that, you can observe very important thing, that is, in these cells you have what? You have proper arrangement and the proper shape in these cells, polygonal shape in these cells, and the cells have a thick boundary. We will think about that, what is that boundary. And the other thing is, these cells are having firm attachments, and there are no gaps between the cells, as well as the cells are also already having a central part, kind of a sac-like part. We will learn in the future, that's called the vacuole, that part. So you can observe these structures from the light microscope under mid power.